Welcome, everybody, to the second half of our exploration of Chapter 2, The Chemistry of Life. And to remind you of where we left off and where we are headed, in the previous lecture video, we covered some of the basic fundamentals of chemistry, including what are atoms, what are ions, isotopes, and molecules. We discussed why water is such an important molecule for living organisms, and we discussed what acids and bases are. Now, in this lecture, we are going to be focusing on answering the question, what are living things made of on a chemical level? And this gets back to one of the 7 characteristics of living organisms that we looked at in Chapter 1. Alongside regulation, evolution, response to stimuli, etc., one of those characteristics was order. Living things have a very specific organization to them. And looking across the diversity of living things, organisms might appear quite different from each other on the surface, like a tiny microscopic bacteria may seem to share not much in common with an elephant, for example, on a superficial level. But on a fundamental chemical level, all living things are actually composed of the same types of molecules. And that's what we're investigating in this lecture. On a molecular level, what makes up living organisms? And you're actually probably more familiar with the answer to this question than you might think. If you just take a moment and consider what you know about what's in your food, this gives you a window into what living things are made of. Because actually, everything that we eat, with the exception of minerals like salt, everything we eat was once part of a living organism, whether that's fruits or vegetables, all different kinds of meat, milk, eggs, all of these things came from living organisms. So, what is in your food? That is what living organisms are made of. You may also be familiar with this little thing called a nutrition label. And because of our existing familiarity with it, we're going to use this as the starting point of our discussion. So living organisms are mainly composed of 4 types of macromolecules. They're called macromolecules because as far as molecules go, they are very large. Macro means big. And those 4 macromolecules are carbohydrates, lipids, proteins, and nucleic acids. And on the left side of the slide, you can get a general idea of what each of these molecules looks like, but we'll talk about their structure in more detail in just a second. Out of these 4 macromolecules, you can see 3 of them represented on a standard nutrition label that you would find on one of your food items. We can see fats, and fats is the everyday language term for lipids. We can see carbohydrates, and we can also see proteins highlighted here in the label. Now, nucleic acids, which is the 4th type of biological macromolecule, they're not on here because nucleic acids essentially means DNA. DNA is found in all living organisms, and because everything that you eat was formerly alive, you eat a significant amount of DNA in your diet every day. However, it has virtually no nutritional value, and because of this, you won't see nucleic acids on the nutrition label. But we will still be learning about nucleic acids even though they aren't on the nutrition label. So, we're going to look at all 4 of these macromolecules one by one, discuss what their molecular structure is, and also what role they play in living organisms. And as we dive into this discussion, one thing to keep in mind is that each of these macromolecules is made up of smaller parts. And those smaller parts are called monomers. Monomers are single small molecules that can be combined with other similar small molecules to create larger macromolecules. So we can think about monomers as being like a single Lego brick. And if you gather a bunch of these Lego brick units and you put them together, then you get the macromolecule. So macromolecules are large molecules that are made of smaller repeating units. Another important thing to keep in mind as we are learning about these macromolecules, and specifically the nutritional significance of each macromolecule, is that nutrition research is notoriously unreliable. And this is the result of two reasons. One is that most nutrition studies are observational, not experimental. Now, let me just pause here for a minute and remind you of what we're talking about here when we say observational versus experimental, because this is something that we learned back in chapter 1. So we learned in chapter 1 about a number of different conditions that affect the quality of a study. And one of those was that an observational study is one where a researcher observes but does not intervene, doesn't set up specific conditions that they're testing. And because of this, it can be very difficult to control the impact of other variables on the outcome of the study. So experimental studies do intervene and set up specific conditions. And therefore, experimental studies tend to produce more reliable results than observational studies. Experimental studies also usually set up a control where you can use that as a comparison to the experimental conditions. So there's a lower chance of unanticipated variables affecting the outcome. We also learned that studies with large sample sizes are better than ones with smaller sizes because with more data, you get a better picture of what's being studied. And we learned that studies with well-balanced samples that have a good balanced representation of participants are better than ones that have skewed samples. And we learned that it's better for researchers to have no conflict of interest rather than having a conflict of interest that might influence them to come to a certain conclusion. So, now that we've reviewed that, getting back to nutrition research, most nutrition research is observational in nature, meaning that there's no control group to compare it to. So let's imagine you were performing a study to ask the question, what effect does lowering your fried food consumption have on cholesterol levels? So under perfect ideal conditions, a researcher would set up an experiment where they have some people cut out fried food and others maintain their normal diet that includes fried food. And then they would compare them. But a more common approach to answering this question would be that the nutrition researchers wouldn't actually set up an experiment, but instead they would just observe by simply asking a group of people how much fried food they're already eating and then comparing the cholesterol levels of the people who are already eating a lot of fried food to the cholesterol levels of people who are not already eating a lot of fried food as part of their normal diet. One major problem with this type of setup is that people tend to be very bad at remembering exactly what they ate and accurately reporting it. So, researchers get a lot of inaccurate data for observational studies like this because people simply don't remember, for example, how many times they ate a salad last month or how many times they ate fast food last week. And also, even when experimental studies are conducted, where the researcher intervenes and says, we're going to have this group of people eat more fried food and this group of people eat less fried food, people are very bad at sticking to those diets. And a lot of cheating goes on because people don't really want to stick to the diet that the researchers are requiring. So, because of these weaknesses, the best and most reliable nutrition studies are ones called metabolic ward studies. And they're called metabolic ward studies because in these studies, participants agree to live in a ward for a period of time. And a ward just means a research housing complex. And in this environment, the researchers can truly control and measure the amount and type of food that people are consuming because they are the only ones supplying the meals. And with that, they can have a very tight, reliable experimental setup. But not surprisingly, metabolic ward studies are also extremely expensive to conduct, so they're also much less common than other types of nutritional research. Um, Obviously, it costs a lot more to put someone up in a ward for 2 months rather than to just email them a questionnaire asking them what they already eat. However, despite being less common, metabolic ward studies are considered the gold standard in nutrition research. And if you are looking for nutrition information to inform your own life choices, these are the studies that you should pay most attention to. The other factor that can make nutrition research unreliable is conflicts of interest. Conflicts of interest are not uncommon in nutrition studies. One recent analysis found that 13.4% of nutrition research is funded by companies in the food industry, which is, of course, problematic because these food companies may choose to selectively fund researchers who have track records of coming to favorable conclusions, or researchers who receive this funding may feel pressured and biased toward a certain conclusion because they feel like they owe the company that helped fund them. One analysis found that there is a stark difference between the conclusions of studies that are funded by the food industry and the ones that aren't funded by the food industry. Specifically, when studies are funded by the food industry, there is a 55.6% chance that they will come to a conclusion that is good for the company, whereas there's only a 9.7% chance of coming to a conclusion that's good for the company when it's not funded by the industry. For example, studies that were funded by the food industry were found to be 5 times more likely to conclude that sugary beverages do not cause weight gain as compared to studies that are not funded by the food industry. So this is another important thing to be especially mindful of when looking at nutrition research and to weigh these conflicts of interest when you're deciding what to consider reliable and compelling evidence for choices that you make in your own life. So to that point, let's flex those critical thinking skills in our first checkpoint of the lecture. So what you see here is a screen clipping of a study entitled Associations of Dietary Cholesterol or Egg Consumption with Incident Cardiovascular Disease and Mortality. This was an observational study of 29,615 participants. 45% of them were men and 55% were women. And how the study worked was that in 1985, the participants were asked to fill out a questionnaire, a survey about their recent dietary habits, basically what they had been eating in the past year. And then researchers tracked their health outcomes for an average of 17 and a half years for each participant, meaning that they continued to check up on each participant periodically for 17 and a half years to see what health issues they had developed. Although at no point during the course of those 17 and a half years did the researchers give the participants another questionnaire about their dietary habits. There was only one questionnaire at the beginning. At the end of the study, the researchers concluded that eating 1 or more eggs per day slightly increases the risk of cardiovascular disease for men. Also, the authors reported receiving consulting fees and funding from multiple drug companies that make and sell cholesterol medications. So, in this checkpoint, given that summary of the study, I'd like you to tell me what is one strength of the study, what's one feature that is good about it, and explain why this feature makes it more reliable. And in the second checkpoint, I'd like you to tell me what's one weakness of the study and why that weakness makes it less reliable. So the bottom line that I'd like you to take away from this when it comes to assessing nutrition information is that it's important to use your scientific literacy skills when you're looking at information coming out of this field. Don't take things at face value. Instead, dig deeper and be mindful of the strengths and weaknesses of any given study. Studies are rarely black and white, good or bad, so it's important to know this when you're weighing how seriously to take a piece of information. And then lastly, be wary of headlines that you see in the media that say things like drinking wine reduces your risk of breast cancer or people who eat chocolate are twice as likely to be obese, because these are almost certainly an oversimplification of what actually happened. It's probably an observational study, and it may not give us the full picture of what's going on behind the scenes of that piece of research. Okay, that being said, let's get into now what is in your food, the 4 biological macromolecules. So the first molecule that we're going to look at is carbohydrates. There are many different types of carbohydrates found in nature. Some of them supply energy that is easy to access for living cells. Others play a structural role and physically make up certain parts of the cell. The monomers, the building blocks of carbohydrates, are called monosaccharides. And that's the scientific name, but the common name that you know them by is sugars. And you can recognize the names of specific sugars because their names always end in OSE, ose. So here's one example you've probably heard of before, fructose. And you can see the chemical structure of fructose here on the slide. This is the molecule that is found in high fructose corn syrup. So you can see that it has sort of a pentagon ring shape composed mostly of carbon atoms in the ring. They're each covalently bonded to each other. So each one of these little lines that you see represents a covalent bond between two atoms. And then there are hydrogen and oxygen atoms scattered, attached around that ring. And so I've just name dropped a term that we learned about back in chapter 2. So we learned about this in the first part of chapter 2. We learned about covalent bonds. So this is a good stage to refresh our memories. And in this checkpoint, I'd like you to answer the question, what is a covalent bond and how is it different from an ionic bond? Okay, so we've seen one example here of a monosaccharide. This is fructose, but there are many different monosaccharides out there. Another example is glucose, which you also may have heard of. You can see that the molecular structure is slightly different, but it's still a play on this same idea of being a ring that is mostly made of carbon atoms, and it's surrounded by branches of oxygen and hydrogen atoms. And keep in mind that what you're seeing here, this in itself is not a carbohydrate. This is the building block of carbohydrates. To get a carbohydrate, you have to put several of these together. So let's take a look at what that looks like. So here we have two monosaccharides, two glucose molecules side by side. And these two can form a covalent bond to each other, like so. And when they do, we're no longer looking at two monosaccharides, we're looking at one disaccharide. A disaccharide is formed when two monosaccharides bond to each other. The prefix mono means one, and the prefix di means two. So a monosaccharide is one monomer, a disaccharide is two monomers put together. And this particular disaccharide that you see here is called maltose. But just like there are many different monosaccharides, there are also many different disaccharides. Maltose is the disaccharide that you get when you put two glucose molecules together. And the common name for this is malt sugar. And this can be found in seeds, grains, and fruits. Lactose is another disaccharide that you get when you put together the two monosaccharides glucose and galactose. And this is called milk sugar. It's the main sugar that you find in milk and dairy. Sucrose is the disaccharide that you get when you put glucose and fructose together. And this is your common white table sugar. So in the biological world, it's found in fruits, veggies, and nuts. So we've seen what happens when two monosaccharides join together and become a disaccharide, but the process doesn't have to stop there. We can keep adding more monosaccharides to this chain and building out this molecule. And when that happens, the molecule is no longer considered a disaccharide, but now it is considered a polysaccharide. The prefix poly means many. And a polysaccharide is what you get when three or more monosaccharides bond to each other. And now what we have is considered a carbohydrate. Polysaccharides are synonymous with complex carbohydrates. To round out this discussion of carbohydrates, let's take a look at a few specific examples of carbs in nature. Starch is a carbohydrate that you've probably heard of before. You can see the actual molecular structure of starch up here at the top right of the slide. This carbohydrate is what plants use for storing sugars for extended periods of time. It's often located in the plant's fruit or its roots or its seeds. So for example, you may associate starch with potatoes, which are a root vegetable. Another example of a carbohydrate is cellulose. Cellulose is a carbohydrate that serves a structural role. It provides support to plant cells, and it's a major component of wood and paper, which are of course made of trees. Glycogen is another carbohydrate. Glycogen sort of is to animals what starch is to plants. Whereas starch allows plants to store sugars in a long-term fashion, glycogen allows animals, including humans, to store these linked sugars for long-term use. It's primarily found in the human liver, but also in muscle cells. And then the last example I have here is chitin. It's spelled like chitin, but it's pronounced chitin. Chitin is to fungus what cellulose is to plants. Chitin provides structural support to fungal cells, and it also plays an important role in insects as well. It makes up their exoskeletons. So if you've ever stepped on a cockroach and you've felt and heard that crunch under your shoe, that is the carbohydrate chitin that's being crushed underneath your shoe there. Now, before we close the door on carbohydrates completely, it's worthwhile returning to the nutrition label, because on this label, you will see that there's more than just total carbohydrates listed. Underneath the category of total carbohydrates, there are a few other things. So let's talk about what those other things are. One of them is dietary fiber. So dietary fiber consists of carbohydrates that your body cannot break. For example, cellulose, that structural component of plant cells, that is a carbohydrate that our bodies cannot break down and derive any nutrition from. So whenever you eat a plant, you are eating cellulose, but you can't digest it, and so it ends up passing right through your digestive tract and out through the other end. So fibers like cellulose are a major component of your stool, and they contribute to your bowel health by keeping your stool healthy. We also see under the carbohydrates category that there are total sugars and added sugars listed. These lines on the nutrition label are not talking about these more complex carbohydrate molecules, but rather the monomers of carbohydrates, the sugars, those single monosaccharide molecules. And so sugars is housed under carbohydrates on the nutrition label because on a molecular level, they are closely related. And so the total carbohydrate that you see in bold actually includes the total sugars wrapped up into it, along with the more complex macromolecule carbohydrates. Total sugar is the total amount of sugar found in a serving, and this includes both the sugar that is naturally found in the food, as well as any sugars that were added after the fact. Added sugars is just the amount of sugar that was added beyond what is naturally occurring in the ingredients. So for example, if we're talking about a serving of orange juice, there is naturally occurring sugar in orange juice that comes from the oranges. But then the manufacturer probably also added some additional high fructose corn syrup or other source of pure sugar to make it even sweeter. So the total sugar would include both of these, but the added sugar would just focus on the high fructose corn syrup, for example. And while we're on the topic of sweeteners, it's a great time to take a look at one of the most common artificial sweeteners in a checkpoint. Sucralose is the name of the artificial sweetener that is found in Splenda. And it interacts with your sweet taste receptors in your mouth in a very similar way that the sugar sucrose does. But your body cannot digest sucralose, so you don't derive any calories from it. And here you can see the chemical structure of sucralose. And based on what you've seen as we learned about carbohydrates, would you classify sucralose as a monosaccharide, disaccharide, or polysaccharide? And explain how you can tell. So we're wrapped up with carbohydrates now. This brings us to the second of the 4 macromolecules. So we're moving on to talk about lipids. And as a reminder, lipids is the scientific term for fats. So when we see fat on the nutrition label, to us that should mean the same thing as lipids. In living things, lipids serve the purpose of providing long-term energy storage and also making up the structure of cell membranes. As a macromolecule, like carbohydrates, they are made of smaller building blocks. But in the case of lipids, there are actually 2 building blocks that go into their structure. One is called glycerol, and the other is called fatty acid. So a glycerol is the one that you see on the left. It's a chain of 3 carbon atoms attached to 3 oxygen atoms, and each of those is attached to surrounding hydrogen atoms. And then fatty acids are chains of carbon atoms, and the number of carbon atoms in the chain, so the length of that chain, actually varies. But it's carbon atoms attached to a hydrogen atoms, and then at one end of the chain, there's something called a carboxylic acid group, which is an arrangement of carbon and oxygen atoms that look like this. This particular fatty acid is called caproic acid, and it's one of the ones that you would find in the fats that are found in goat's milk and goat meat. So there are many different fatty acids out there. Each have a different number of carbon atoms in their chain, and they have different names, and they can be found in different sources in nature. Here's another example. This one, as you can see, is longer than the one that we saw in the previous slide. This one is called lauric acid, and lauric acid is found in coconut butter. And we're going to use lauric acid as an example to see how these monomers come together to form a lipid. So here is our other monomer. Here's the glycerol molecule. And each one of these 3 oxygen and hydrogen pairs on the glycerol molecule serves as a sort of docking station for a fatty acid molecule. So we have not 1, not 2, but actually 3 fatty acids that will come in and they will form a bond with the glycerol molecule. And this lipid we would refer to as a triglyceride, with the prefix tri meaning 3, because there's 3 fatty acids attached to the glycerol. There's another type of lipid that is a variation on the triglyceride structure, and we're going to talk about that in just a minute. But before we do, one quality of lipids that is important for us to address is the fact that they are hydrophobic. Hydrophobic literally means fearing water, but it is the scientific term that means they don't want to mingle with water. They want to remain separate from water. And we talked a little bit about this in the first part of chapter 2. You may recall this animation showing vegetable oil, which is a lipid, in the process of forming a separate layer with the water in the glass. And the fact that lipids are hydrophobic is actually very important for living organisms, and we'll talk about why that is in the next chapter. But to set us up for that, in this chapter, we are going to answer the question, why are lipids hydrophobic? What about their chemistry makes them hate water? Well, you may remember from our previous lecture that there are 2 types of covalent bonds, polar and nonpolar. Nonpolar covalent bonds are formed when 2 atoms share their electrons equally because they have similar levels of electron greediness. And it turns out that this is the case for carbon and hydrogen. Hydrogen and carbon will each happily bring an electron to the table, and they will share them equally with each other. And in doing so, they create a nonpolar bond. They do not develop slightly positive and negative ends that would result from unequal electron sharing. We saw how that happened in water. It does not happen with hydrogen and carbon. Instead, they form a nonpolar bond where they share their electrons equally. So they maintain equal sharing with their arrangement. Now, why are we dwelling on the relationship between carbon and hydrogen so much? Well, the fatty acid chains of the lipid are made entirely of carbon atoms bonded to hydrogen atoms. So we have these three very long, significant nonpolar chains of atoms here. Now stay with me here on this review. Do we remember this slide? So in the previous lecture, we learned that one of the properties of water molecules is that they can easily dissolve ionic and polar molecules. Hopefully you remember this picture showing a collection of water molecules and dropping a molecule of sodium chloride table salt into there. And this works because sodium chloride is an ionic molecule that has electrical charges in it. So the attractions that are found between water molecules, between the positive end and the negative end of separate water molecules, they can be substituted for attractions to other molecules that have full or partial electric charges. So here the sodium ion fits right in because it has a positive charge and it's attracted to the negatively charged ends of the water molecules. The chlorine ion fits right in because it has a negatively charged, it's negatively charged and it's attracted to the positively charged ends of the water molecules. So when you put this ionic molecule into water, it fits right in. It's one of the crew. Conversely, if you take a nonpolar molecule and drop that into a sample of water, it doesn't fit within this network of attractions. It's not attracted to the water molecules aren't attracted to it, and so it gets pushed into a separate layer. And that's why lipids, like oil, become separated from water, because those many, many nonpolar carbon-hydrogen bonds within the fatty acid chains are responsible. So now that we've established why lipids are hydrophobic, we can introduce the variation on the triglyceride that I previously mentioned. So triglycerides have a glycerol molecule bonded to 3 fatty acid chains. But there is another type of lipid called a phospholipid that has a similar structure, save for one feature. Instead of having 3 fatty acids, phospholipids only have 2. And in the place of the third, there is a different structure locked into that third site on the glycerol molecule. And this structure is called a phosphate group. It's named for the fact that it has an atom of the element phosphorus situated in the center. And now the thing about this phosphate group is that the bonds found within it, the bonds between the phosphorus in the middle and the oxygen atoms all around it, those bonds are quite polar. So we have this polar component over here, and meanwhile, the 2 remaining fatty acids continue to be nonpolar. And now just a quick tangent here. The way that these phospholipids are typically drawn when biologists are illustrating them in a simplified way is not with the full detail that you see here in this image where it shows the placement of every single one of the atoms. So instead, the illustration of the phospholipid gets significantly simplified down. Biologists draw the polar phosphate group as a circle at the top, and then the 2 fatty acid chains as these tails that are hanging off of the circle. So when you see this drawing that looks kind of like this weird little jellyfish, it's actually illustrating a phospholipid, but it's not showing the full complexity. Okay, so getting back to the chemistry of the phospholipid. The phospholipid has this unique duality about it, in that one end of the molecule has a polar structure, the phosphate group, that dissolves well in water and wants to be around water. So the phosphate head is hydrophilic. That's the term that means water-loving. It wants to mingle with water. The other end of the molecule, these fatty acid tails, consists of nonpolar bonds that are hydrophobic. So water wants nothing to do with them. They don't mingle well with water. And they're attached to each other. They're covalently bonded. The hydrophilic and hydrophobic part are part of the same molecule. And as I said before, this feature, this quality, is actually going to be really important for cell formation. And we'll talk about that in our next chapter. But for now, I want you guys to flex your imaginations a little bit by answering this checkpoint question. What would you expect to happen to phospholipids that are placed in water? What would happen when you put a molecule that is partly hydrophobic and partly hydrophilic in water? And here I'm not necessarily looking for the right answer. I'm just looking for an answer that shows evidence of critical thinking and coming to a logical conclusion that you can explain. And hold this question in your brain because, again, next week when we talk about cells, we're going to revisit it and see what actually happens when you drop phospholipids into water, and you'll be able to compare your expectations to what we really see in nature. So again, before we leave lipids behind, there are some other items from this nutrition label that we need to touch on that fall under the category of fats. So one of the things you see under the fat line on the nutrition label is a line for saturated fats. There are two major classifications of fats. There's saturated fats and unsaturated fats. Unsaturated fats are ones that contain kinks in their fatty acid tails, whereas saturated fats are ones that do not contain any kinks. So let's see what this looks like. Here we have a saturated fatty acid on the left, and on the right we have an unsaturated fatty acid. Now, I know that here in these images, which were taken from your textbook, it's showing the chain in a zigzag line rather than a straight line for both of these, but those zigzags are not the kinks that we're talking about. What you see on the right-hand side, where the fatty acid chain makes a sharp turn upward, that is a major kink in the fatty acid chain. And that's the distinction between saturated and unsaturated fats. And this difference in their chemical structure actually manifests in a way that we can observe in our everyday lives. In saturated fats, because their fatty acid tails lie more flat, the molecules pack more tightly against each other, and these fats are typically solid at room temperature. In unsaturated fats, on the other hand, the kinks in the fatty acid tails mean that the molecules aren't able to pack together as tightly, and they have a looser structure that tends to be liquid at room temperature. The difference in the chemical structure also has consequences for our health when we consume these different types of fats in our diet. So most research indicates that saturated fats contribute to higher levels of unhealthy cholesterol, also the formation of plaque in arteries, which leads to a higher risk of cardiovascular disease. And then conversely, research indicates that unsaturated fats tend to reduce blood cholesterol levels and improve cardiovascular health. And these findings are based on metabolic ward studies, which, remember, are those high-quality experiments that take place in a very controlled and very reliable setting. The other thing that you will see on the nutrition label under the category of fats is trans fats. Trans fats are a type of unsaturated fat that has had the kinks in the tail forcibly, artificially removed. So trans fats were not actually a significant part of the human diet until a food manufacturing process called hydrogenation was invented. Hydrogenation is a chemical process that artificially turns liquid oils into solid fats. So when you see items in the grocery store like Crisco, and it says that it's made with vegetables, or margarine products like I can't believe it's not butter, these are solid fats that were created from liquid oils through the manufacturing process called hydrogenation. And as a part of that process, some percentage of the unsaturated fats accidentally get turned into trans fats. Early on in the history of these hydrogenated products, the health consequences of trans fats were not known, but over time, research has shown that they are very bad for our health. Higher trans fat consumption leads to an increased risk for obesity, high blood pressure, cardiovascular disease, and diabetes. And the effects are so significant that trans fats have been banned in a lot of places, meaning that in most hydrogenated products that you can buy in the store today, like Crisco on the shelf today, the trans fats have actually been removed before they sell it to you. So let's tie up our discussion of lipids and fats with a checkpoint. In this checkpoint, I would like you to take the following fats and sort them into the categories of saturated and unsaturated fats. We have butter, olive oil, coconut butter, lard, vegetable oil, and steak fat. So we're closing out lipids now, and the next macromolecule on our list is the proteins. Proteins are extremely important. They are arguably the most important macromolecule because they are involved in virtually every function that happens in virtually every living cell. Proteins are responsible for transporting materials, for making up important structures in cells and in the body. They're responsible for the regulation of functions that maintain our homeostasis. They protect cells from threats and invaders. They communicate and send signals between cells, and so much more. So without proteins, life as we know it would be completely impossible. The building blocks that proteins are made of are called amino acids. And amino acids have a chemical structure that starts with a central carbon atom that is attached to 4 separate things. The first is an amino group. That's what we call this collection of a nitrogen atom attached to 3 hydrogen atoms on the left. The second thing is a carboxylic acid group, which we saw before when we looked at lipids. This is the same thing. And then there's a hydrogen atom. And the fourth thing is called an R group. The R group varies in its structure because there isn't just one amino acid, there are many. And the thing that makes them different from each other is the structure of the R group. So let's take a look at a few specific examples of amino acids and their R groups. So this amino acid is called serine. Its R group is made up of a carbon attached to 2 hydrogens and an oxygen, and then the oxygen is attached to another hydrogen. Here's another one. This is glycine. Glycine has the smallest and simplest R group amino acid. It's just a hydrogen atom. And then the third one you see on the screen is called aspartic acid. It has an R group that starts with a carbon bonded to 2 hydrogens, and then it ends with another one of those carboxylic acid groups. So these are just 3 examples, but there are actually 20 amino acids that occur commonly in most living organisms. And here they are, all laid out in a chart. You can see that their R groups have been highlighted for you in a darker color. And now I do not expect you to know or memorize all of their structures, but what I do want to point out to you is that biologists classify these 20 amino acids into several different groups based on the chemical nature of their R group. So these categories are labeled and color-coded in this image. We have the nonpolar amino acids, which means that their R groups are composed of nonpolar covalent bonds. The atoms in the R group are sharing electrons nicely with each other. We have the polar amino acids, which are ones where the chemical structure of the R group contains polar covalent bonds. And then there are electrically charged amino acids as well. There's ones that have actual ions within their R group. They have atoms that have a full-blown negative charge or a positive charge because they've either lost or gained an electron from another molecule. So there are 2 amino acids that have a negative ionic charge, and that's shown in that purple color. And then there's 3 that have a positive electric charge, shown in green. So once again, these amino acids that we're talking about, these are just the monomers. They're just the building blocks of the protein. In order to make a protein, you have to put these monomers together. So here we have the amino acids, aspartic acid and glycine. These 2 can become bonded to each other, and now we're on our way to forming a protein. And just to help you orient yourself now to the larger molecule, here are the 2 R groups of the amino acid. I put a box around each of them. And just like we saw when we were building a carbohydrate, the process will keep going and the chain will keep lengthening. So here we're bringing in the amino acid serine, which will form a bond to the existing chain and lengthen it even more. And here are those R groups again. And this will continue on and on and on, dozens, hundreds, even thousands of times to make an extended chain of amino acids that we call a protein. So here we have a highly simplified depiction of what that big long chain of amino acids might look like. In this illustration, each amino acid has been represented by one little bead in the chain, and we want to sort of take this broad overarching view of looking at the protein chain like this, because the key to how any given protein works in the body is the way that the chain becomes folded into a three-dimensional shape. The chain won't stay a chain. It will bend, it will fold, it will wind, it will cluster on itself to form a distinct shape. Every protein has a distinct shape, and what drives the formation of the shape from the chain is attractions that individual amino acids within the chain have towards each other. So some amino acids, namely the polar ones, will be drawn toward each other. Others will be repelled by each other. So for example, if you get two amino acids in the chain that both have a positive electric charge, they want to push away from each other. So those attractions and repulsions dictate the folding process. And then the final shape of the protein dictates its function. And now I'm going to say that again because this is a very important point that will come up again later when we're learning about genetic diseases. The shape of the protein is what dictates its function. And to really drive this point home, I'm going to give you a few examples. So hemoglobin is the name of the protein that is responsible for carrying oxygen from your lungs through your bloodstream and to all of your organs and tissues in your body. And the shape of hemoglobin, which is shown here in red, is such that it has a little pocket in the middle. It has this slot where another molecule called heme is able to fit into that slot. And then the heme molecule is what grabs and holds the oxygen. So we can see here how the shape of hemoglobin is related to its function and the role that it plays. Another example is GLUT4. GLUT4 is a protein that is actually embedded and buried in the membranes of our cells. It's like a tube or a channel that allows glucose sugar molecules to pass through the membrane and to the inside of the cell. And then once inside the cell, the sugars can be broken down and used for energy. So again, the channel-like structure of the protein dictates its function. And then a third example is antibodies. Antibodies are proteins that are made by your immune system. And what antibodies do is they attach to invaders, such as bacteria or viruses or parasites, and they flag them for destruction. So every antibody is a protein that is tailor-made with a binding site that's very specific so that it matches and fits a specific virus or bacteria. So once again, the structure of that protein is critical for it being able to do its job. One final type of protein that I want to highlight and give special attention to is proteins that serve as enzymes. One example is a protein called amylase, which is found in your saliva. So as you're chewing, amylase starts the process of breaking down the carbohydrate starch so that when it hits your stomach, it's already on its way to digestion. And this breaking down of starch is a chemical reaction. And as a general rule, enzymes are proteins that facilitate chemical reactions in living organisms. All of the chemical reactions in living organisms. They build things, they break down things, everything that's needed by our cells, all of the structures in our bodies, they are the result of chemical reactions that are made possible by enzymes, which are proteins. One final thing I'd like to note about proteins that is something that's not actually broken down for us on the nutrition label, but it's still a good thing to know, is the distinction between essential and non-essential amino acids. While all 20 amino acids are important components of proteins, some of them can actually be created by our bodies, while other ones can't be. Specifically, there are 9 amino acids that our bodies cannot create. And so the only way to get these amino acids is by eating them. And because of this, they are called essential amino acids because they are essential to get them in our diet. And I've listed those 9 essential amino acids here in the green box. The other 11 amino acids are ones that our bodies can make, and so these are called non-essential amino acids. Those are the ones in the blue box. And as we finish off our discussion of proteins, I have a checkpoint for you pertaining to essential and non-essential amino acids. Some foods have a large quantity of protein in them, but they also have a little note that says not a significant source of protein next to the protein line on the nutrition label. So pork rinds, which is what you see in this image here, also called chicharrones, are one of these. And this is fried pig skin. And pig skin is made primarily of the protein collagen. So if you look at the label, you can see that they have lots of protein. They have 11 grams of protein, but at the same time, just to the right of the protein on the label, it says they're not a significant source of protein. So what's the deal here? Well, this table at the right breaks down the percent representation of the essential amino acids in collagen, which is the main protein found in pig skin. So the leftmost column in the table lists the essential amino acids, and then the rightmost column shows the percentage of those amino acids found in collagen. And then the middle column, to serve as a point of comparison, shows the percent of each essential amino acid found in milk protein as well. So looking at the top row, we see that isoleucine, the amino acid isoleucine, is 8.7% of milk protein. That means that out of all of the amino acids in milk protein, 8.7% of them are isoleucine. So, after viewing this table, why do you think pork rinds have this little label where they aren't considered a significant source of protein, even though they have a whole lot of protein in them? The fourth and final macromolecule to discuss is the nucleic acids. Now, again, you'll notice that nucleic acids are not highlighted on the nutrition label. They aren't listed because they have no nutritional value, but they are nonetheless found in all of your foods, and they play a very important role in living organisms because nucleic acids serve as the genetic material for living organisms. They carry information encoding what traits an organism should have. There are 2 main types of nucleic acids, DNA and RNA. And we will learn much more about both DNA and its lesser known cousin RNA later in our class. But for now, we just want to cover the basic structure of nucleic acids. The monomers of nucleic acids are called nucleotides. And these monomers have a consistent structure that's made up of 3 parts. Each monomer has a sugar molecule, which is attached to a phosphate group and also attached to a nitrogenous base. And some of that probably sounds familiar to you. So in this checkpoint, I'd like you to tell me, in what other macromolecule in this chapter did we see a phosphate group? And then also, in what other macromolecule did we see sugars? So the building blocks of nucleic acids are called nucleotides. How do we get from the building block to the nucleotide? At this point, you probably won't be surprised to learn that the nucleotides like to form bonds with each other. So here we can see an example of 2 separate nucleotides coming together and forming a unit. And this will carry on until it creates very, very long chains of nucleotides. And those long chains are what we consider the nucleic acid. So to give you some perspective on how long these chains get, the largest human chromosome is made of strands of DNA containing about 250 million nucleotides. And again, we'll go much deeper into DNA later in the semester, but this is where we're going to leave things with nucleic acids for now. To wrap up this lecture, I have just 2 remaining checkpoints for you that should serve to sort of recap the 4 macromolecule groups, both in structure and in function. So first, I'd like you to match the macromolecules with the name of the monomers that compose them. And then secondly, I'd like you to match the macromolecule with one of the possible functions that it serves in living things. And once you finish this checkpoint, you are finished with the second half of chapter 2.